Hello everyone, welcome back to one more session. In this session, I'll be discussing another concept, that is variation and the types of variation. In the previous session, we have discussed the introduction part of heredity and variation. This particular concept is required only for NEET. For board, it is not required actually. So the board students no need to watch this video. Okay. So first of all, what is variation? We know that variation is nothing but it is the degree of difference in characters of an individual. That means the characters in a particular organism differs from its siblings also, and it differs from its parents also. That is actually the variation. Do you think that variation is important? Obviously variation is very important for the evolution course. Sometimes the variation may also lead to the diseases, getting diseases. Good example is cancer actually. Got it? So the due to the mutation of the genes, and that could actually bring the variation of that particular gene, and which produces abnormal protein. Because of all these things, that may lead to cancer or any kind of genetic diseases, okay, which is dangerous. Otherwise actually variation is very good. A slight variation is really good for the evolution. You know that actually, so every, obviously sexual reproduction brings variation, correct? Because actually there is recombination process. You know that during the formation of gametes, male gamete or the female gamete, obviously the meiosis should take place, correct? Why the meiosis? There are two significances of meiosis. One is actually it will bring the shuffling of the genes within the homologous chromosome, number one. The other one is actually, it will reduce the number of chromosomes to half, that is 2n becomes n, correct? So 2, finally ultimately n plus n finally makes 2n in the state, that is diploid zygote, in the zygote actually, okay. So that is actually the significance of meiosis. Now what we have to learn is which particular agent actually causes this variation. Okay, so first to explain that, I'm going to draw the diagram of DNA. Let us assume that this DNA belongs to a particular human being. Okay, so you all know that actually DNA is a double helix model. Okay, it is a stranded structure. Okay, here I will write the sequences, that is the nitrogenous bases. We write AG, CT, AC, G like that, and I'm going to write its complementary base pairs also. Here I'm going to write T, C, G, A, here actually T, G, C like that. We all know that actually that segment of the DNA, if it produces a protein, then that segment of the DNA is called as the gene. If you look into the sequence in the DNA, obviously like it is exactly same in all the human beings. That means 99.9% of the sequences are exactly same in all the human beings. Only 0.1% differs. That's what which makes us different individuals. Okay, 99.9% of the sequence is exactly same in our DNA. Only 0.1% differs. Okay, so here let us assume that this particular segment of the DNA is a gene which produces a protein. Let us assume that it produces a protein. Actually, this protein is responsible for certain physiological activity in our body. Okay. Now, this is actually a normal gene which produces a normal working protein which actually responsible for physiological activity or maybe structural support also. Now, due to certain mutation, the mutation is nothing but change in the base pair. You could see the reading sequence G, C, T, A, C, G is the reading sequence of that gene. Okay, let us just assume that instead of C here, supposed to be here, C should be here, but instead of that, another sequence, another nitrogenous base comes. Let us take A here. It should not be here, but it is there due to mutation. It could be because of ultraviolet radiation, or it could be because of any chemical agents, for example the cigarette smoking or anything, or maybe the habits, lifestyles, and or maybe like genetics, whatever it is. Okay, or it could be because of the virus. This is now changed. Okay, so as a result, C and G base pair is now replaced by A and T. Now this particular gene produces an abnormal protein, okay, because that gene is mutated. As a result of the abnormal protein, the physiological activity is affected, and as a result it could cause diseases. And see, if there is any change in the sequence of the DNA due to certain habits or due to the lifestyle or maybe environmental factor or maybe like the natural process, that actually leads to variation actually. Got it? So variation. Okay. The variation sometimes it is good, sometimes actually really bad. Okay, so that is actually one thing. So this is reason number one for the variation. It could occur at any moment in our life. At any moment in our life, actually, remember it. The second one is, so we all know that actually, so this is the chromosome. Okay, so maybe due to the deletion, we know that actually the genes will be there. Correct, the genes will be there. That is actually the place where a gene is situated. That we call it as locus. In the locus, the genes are situated within the chromosome. Okay, so let us assume that due to certain agents, okay, this particular chromatid is deleted. A particular piece or segment of the chromatid is now deleted. It's gone actually. Okay, that means actually here the 3 genes are lost. Correct, due to the deletion of the chromatid, 3 genes are lost. Now assume that if it is present in the gamete, obviously it's going to form what you call the offspring because of the fertilization process, and obviously the particular offspring will have this chromosome which will bring the variation. Correct, that means actually he may suffer from certain diseases. That is variation. Correct, so this is the second reason. What is the second reason? The chromosomal aberration, that is the deletion in the chromatid. Deletion happens. As a result of the deletion, obviously some genes will be lost in the offspring. Okay, the third one is, so we all know that the chromosomes are always occur in pairs. We call it as homologous chromosome. Okay, so this is the homologous chromosome. Okay, so we all know that during the formation of any gametes, it could be sperm or ovum. In this case, I am taking sperm, okay, as example. So we know that this particular homologous chromosome should separate. One chromosome will go into one gamete. This another chromosome will go into the another gamete, like that. So 2n becomes n, correct, in each gamete. So, but sometimes because of certain factors, these 2 homologous chromosomes fails to separate, and as a result, what you see is the homologous chromosome, both the homologous chromosome now enter into one sperm. Here, this sperm will not get that homologous chromosome. And suppose if this sperm actually fertilizes a normal ovum, okay, which containing around 23 chromosomes. Here actually how many chromosomes? Obviously 23 plus 1, right? Because of this case. So 23 plus 1, 24 chromosomes. So 24 plus 23 during fertilization, it makes 47 chromosomes in the offspring. Is it abnormal? Obviously it is abnormal. Can we call this as variation? Yes, it is variation. So because of 3 reasons, the variation occurs. Okay. Now, there are 2 types of variation. Particularly, you could see here I have written somatic variation. Very simple actually. Let us assume that this is male and this is female. Okay. And both are actually, are husband and wife. They are actually married. Let us assume that, both are normal. Both are normal actually. They don't have any diseases. They are normal. Okay. They are going to produce the young one. So, this is actually the young one. Now, because of his lifestyle habits, okay, or maybe because of the environmental factors, it could be anything, okay, he gets a disease, that is cancer. Somewhere else in his lifetime, he gets actually cancer. Why? Because of the lifestyle, or it could be because of the environmental factor, physical, chemical or biological factors which affected mutation in its DNA or gene. As a result, he actually gets cancer. But it is completely absent in his parents, obviously. Okay, so this is actually variation, right? So variation. Okay, so this particular disease will not go into his son, obviously. If he produces the offspring, obviously he will not suffer. He may suffer from cancer or may not suffer from cancer. Let us assume that he will not get any cancer. So this is actually the thing to remember. That is actually somatic variation. Somatic variation means acquired variation. So that means acquired. It could be because of environmental factors. Okay, so this is the thing to remember. Okay, so here mainly somatic variation is due to environmental factor actually. So it is not hereditary or inheritable. It is not inheritable actually. Got it? So it can get mutated. The genes can get mutated at any stage of the life. Got it? And that same gene will not get inherited to the offspring actually. Got it? So that is called a somatic variation. Whereas germinal variation, so in this case, it's mainly like there are 2 types. One is actually continuous, other one is discontinuous. So germinal variation is nothing but it is also called as inheritable variation. Heritable. That means actually the variation which actually mainly because of the heredity. That means actually the characters which are actually transferred from one generation to the other generation. This is actually inheritable variation. Okay. Suppose I will take one example. This is a male and this is a female, that is husband and wife actually. This male is suffering from a disease called as hemophilia. Hemophilia is also called as bleeders disease because of the mutation. We'll talk about it later. It's a genetic disorder. Okay, so they produces an offspring actually, son. The son may, or it's like there is a high possibility that son will get this disease, hemophilia actually. Got it? So, or maybe like the daughter will not get it, but the son will get that disease actually. So that is inheritable, correct? That is actually called as germinal variation. It's mainly because of the sperm or the ovum, the germ cells, obviously because of the mutation within the germ cell, and that is actually transferred to the offspring, and again from the offspring also produces the germ cell, again actually because of the fertilization it will transfer to another, like the third generation, fourth generation, fifth generation, it will go on like that. That is actually called as germinal variation. Okay, there are two types actually. One is continuous, the other one is discontinuous. One thing to remember, that this is hereditary. In this case, it is not hereditary. That's it. Okay. So, in continuous variation, what you see is, for example, let us assume that we take one example here, that is sickle cell anemia. Okay, sickle cell anemia, which is also a genetic disorder. Sickle cell anemia. Okay. So here, same male and female, and they produce the young one. Okay, here suppose the male is suffering from sickle cell anemia, it's a genetic disorder. Here the son also suffer from sickle cell anemia, correct? Because actually there is a mutation in the gene of the father. The same gene actually now entered into the sperm of this. Okay, so during the fertilization, what happened? The same defected gene also enters along with the chromosomes, and fertilization takes place, and as a result, offspring is produced. That offspring which contain a defected gene, which is actually given by the father actually, and again the same gene will travel to the third generation, again the fourth generation. This process will continue. So that means it is continuous. There is no end. Whereas in the case of here, the somatic variation, this is the particular variation acquired only during the lifetime, and that particular character will go away when that person dies, correct? So that is actually the thing. Here, no, it will continue because it is there in the gene. Okay. Next thing is discontinuous. Here it's also same actually, that is heritable variation, inheritable variation, that is germinal variation. In this case, for example, like let us take Down syndrome as one example. In this case, let us assume that this is the male and female. Okay, so female will produce the ovum and this fellow will produce this sperm actually. So as I told you that if the homologous chromosomes fails to separate, as a result what happened actually, the sperm will get extra chromosome, that is instead of 23, the sperm will have 24 chromosomes. So let us assume that he has produced 24 chromosomes. Okay, so here the sperms containing extra chromosome, which is abnormal sperm. If the same sperm actually fertilizes the normal ovum, which is containing 23 chromosome, so that person, the offspring, will have 47 chromosomes. Correct? He will have 47 chromosomes, which is actually variation. But let us assume that this condition is actually leads to a disease called as Down syndrome. I will talk about it later. Down syndrome. The person who is suffering from this offspring is now suffering from Down syndrome because he has one extra chromosome. Okay, marries a normal woman, and as a result they produce the offspring. In this case, the Down syndrome fellow will produce a normal sperm which is having 23 chromosome, and lady with the ovum, that is also 23 chromosome. So total 23 plus 23, during fertilization it becomes 46. That means their offspring will be normal. That means actually this is because of heredity. That is, father has given extra chromosome, okay, due to some fault or mistake or factors, and as a result the chromosome number is increased in the sperm of that father, and that sperm actually fertilizes the young, this one ovum. As a result, the young one is having extra chromosome. That is abnormal, but this will be restricted to only his lifetime. He will not give the same chromosome to his the offspring actually. That means actually it will get restricted. It will get restricted, but we cannot call it a somatic variation, because somatic variation, normal, the disease is acquired only during the lifetime, but in this case it is by the father, heredity. So that is actually discontinuous variation. So that's all about variation.